Welcome, everybody. I'm really pleased to introduce our speaker tonight. Dr. David Zlesak is a professor of horticulture at the University of Wisconsin River Falls. He's been breeding landscape plants for greater adaptability since 1984 and has cultivars of roses, ninebark, heliopsis, and ageratum on the market. He works with the American Rose Trials for Sustainability, Earthkind, and National Clean Plant Network Roses programs, and loves to study resistance to rose diseases, viruses, and rose black spot with colleagues at the University of Minnesota and Texas A&M. David, I'm really pleased that you made the trip over here from River Falls to join us today. He tells me he was up at the U of M today, so he uses time in the Twin Cities well, but we'll listen to his talk on managing disease through genetic resistance, examples of techniques being used with roses. And then we'll have time for questions definitely at the end. So come on up and we look forward to hearing more from you. Thank you.
So it's great to be with all of you here tonight. So I remember years and years ago, when I first came to the group, as a young graduate student, Norma Booty coordinated had me come. That was, 25 years ago, 20 years. So it's really fun coming back and all the memories that I share with so many of you out at Umore Park, all those years that we were able to do those demonstration gardens and have so many wonderful events, and especially treasure the time that we got to spend just out there together, the various gardens, even if nobody else came to visit us. It was great. So thanks for having me here tonight. And so my hope from tonight's topic is to give us some hope, some perspectives towards developing disease-resistant plants for lower maintenance landscapes, how it's done, some examples with what I'm doing with Dr. Stan Hokinson and others at University of Minnesota. So as we think about pest tolerance, disease resistance, adaptability, we want to have plants that are adapted to our region without a whole lot of extra pesticides and additional care. Yeah, there's tender things maybe from zone 5 or 6 that will baby or coddle, but for the most part, we want well-adapted plants, be it roses that are disease resistant, ninebarks, fruit trees, whatever it might be. So these techniques can apply to all of the different types of crops. Thank you. So how many of you have grown roses before? Yeah, how many of you had roses that looked like that? Unique cultivars, varieties, individuals that have the disease resistance to the key regional pests that we face. It's a great asset. So for instance, here, this happens to be the rose double knockout. How many of you have heard of the knockout series, seen them at the store? Maybe there's so much of it that you wish that there was other varieties too, because you already have some. So my childhood mentor, Will Rattler, he bred knockout, and he was the director of Berner Botanical Gardens. I grew up in the Milwaukee area, and Berner has a very historic connection with roses. Jackson and Perkins, how many of you remember the Jackson and Perkins catalog? Okay. So the breeder at Jackson and Perkins from maybe the 30s to the 60s, Eugene Berner, his brother was a landscape architect, and that's what Berner Botanical Gardens is named after. So in Milwaukee, they got all of Eugene's best new roses from New York, where Jackson and Perkins was placed. So Will, as director of the gardens there, had this extensive 3,000 to 4,000 rose collection, this massive garden. And most all those plants needed insulation for winter, preventative fungicide sprays. So beginning in the 1970s, he started to grow above average roses at home, interbreed them at his modest residence, and began to increase disease resistance. Whereas the major breeders in California, where the climate's dry in the summer in the valley and this and that, they didn't have that issue. And the general feeling was better life through pesticides. We could overcome anything. So he had that vision. And he kept pushing it, intermating the above average roses, getting greater and greater disease resistance. And finally in 2000, the original knockout rose came on the market. And it was leaps and bounds above the current rose varieties in terms of disease resistance, because that's what he prioritized. That's what he focused on. So we're a little colder than him in Milwaukee. So sometimes it's a little marginal for winter. But we can have a vision and work towards it, be it ash trees that are resistant to emerald ash borer, chestnut trees that are resistant to chestnut blight, elm trees, American elms that are resistant to Dutch elm disease, like the St. Croix elm that was selected nearby here. So we don't just have to rely on those selected out east like Princeton and other cultivars. So as we overcome or have some individuals that have resistance to these diseases, it's a great asset to us as gardeners or landscapes. So diseases are dynamic. The pathogens, the fungi, the bacteria that lead to these maladies in our plants are always moving and recombining their genes, and some have stronger. Plant pathologists, people that study plant diseases, plant breeders always have a job. There's always a new problem. So our goal is to try to overcome those problems and keep the plants that we love available to us. So as we think about disease caused by a living pathogen, so one thing that I do in my introduction to plant science class, we have one of the labs on diseases and insects. And so after our weekly quiz, I walk around, I try to be creepy, and I ask them, do I put you at ease? And some of them are like, yes, or dis-ease. So as we think about the term disease, that just means that the plant's not growing normally. And we can have abiotic diseases, like when salt from our sidewalk or the roadway interfere with our plants and their normal growth, or biotic, caused by living things like bacteria and fungi. So this disease triangle, and I bet we've all seen the triangle, right? So there's three major sides to that triangle. In order to get disease, that area in the middle, we need a susceptible host, a virulent pathogen, that means a bacteria, fungi that can cause disease on that particular plant, and a conducive environment. And that often means a relatively high humidity environment, a lot of extra moisture. And when all those factors come together, we can get disease. So whatever we can do to try to reduce any of those sides of that triangle, we could reduce the amount of disease or that area of the triangle. So for instance, in the upper right-hand triangle, we could grow a less susceptible host. All right, so a variety that has greater disease resistance. It might not be immune, but it doesn't allow disease to grow on it quite as freely. And then we could reduce the amount of disease that way. We could also grow our plants by shrinking the environmental side. We could grow our plants where there's more air circulation, more sunshine to dry off those leaves earlier in the day. We could also change the environment on the leaf surface by spraying fungicides and other things too. All right, so here's part of my garden at home. And so there's oodles of roses to the left there in the property line. Look at those arborvitaes, and thankfully they're on the north side of the property, so they're not shading my garden. I'm grateful for the windbreak in winter. Somebody planted them 20-some years ago. They're tall and narrow, so they don't take up too much width, but they give me a wonderful windbreak. So it's very open there, and it's very sunny. So it's an environment that's less conducive to disease developing compared to an environment that's quite shady, especially in the morning where the dew lasts even longer. So we can alter the environment to help in terms of right plant, right place. And we can irrigate in ways that reduce disease, of course, too. Drip irrigation, of course, keeps the leaves dry. Or if we do overhead, just because we don't want all that plastic and infrastructure hoses all over, we can do so early in the day, and hopefully that extra moisture will evaporate. So we can also alter the environment on the surface of our plants by spraying and putting chemicals there as well. So the side of the triangle that we don't have a whole lot of control over is the pathogen, what blows in. So as that pathogen blows in, for instance, here's a picture of black spot on roses is caused by a fungus. And there's lots of different variations amongst the different fungal individuals that make up the species that cause this disease, black spot. Just like we're all humans in this room, but there's genetic differences among us. And some of us are better at some things than others and so on. So there's different forms of this fungus that can infect some roses, but not others. So whatever blows in, we don't have a whole lot of control over that. So that side of the triangle, we don't have a whole lot of things we can do. So the main thing that we could do is rake up those fallen leaves that are filled with spores to try to slow down the next cycle of that pathogen and slow its spread a little bit. But that can be labor intensive and challenging. I've been studying black spot with Stan Hokinson and others over the years, over 20 years now. It's been really fun learning more about the fungus, its interaction with roses, with ultimately the goal of developing roses that are resistant to as many different forms of that fungus as possible. And we'll talk about the mechanisms to do so. So first of all, here's a question. What part of the plant, the top, the bottom, the middle, if we're going to get black spot, do we usually see it come in first? The bottom, that's true. That's true. So it comes in from the bottom and it moves up throughout the plant. But the truth is, if you collected leaves from the top, middle, and bottom, and you pulled them off and you put them in little boxes of moist paper towel like we do, and then put fungus on them, the ones at the top are the most susceptible. So why does it start from the bottom? More water, more humidity, it's the environment. It's the environmental side of that triangle. So it's more shaded, it stays more moist. And black spot needs at least 6 hours of free water, dew, extra rain, irrigation, water sitting on the leaves for the spore to be able to germinate and penetrate and get in. So, so that happens more readily within the tight canopy of the plant than up at the top where the wind, the sun will dry it off. What temperature do you think black spot loves best? When you say A, 50, it's a cool season. When you say 60, okay. When you say 70, just like I like my house, yeah. 80, it's gonna get hot. 90, okay. So in controlled experiments, 70 degrees it grows the quickest, but it'll still grow at the other temperatures too. So thankfully, when we're pulling leaves off of the roses and we're bringing them into the labs in these little clear salad boxes of waste paper towel and inoculating with black spot, room temperature is perfect. Perfect. So here's the spores of the fungus that leads to the disease black spot. And the scientific name of the fungus is Diplocarpin rosae. So rosae refers to, okay, it only infects roses. And then diplocarpin, di means what? 2. And look at those spores, there's 2 compartments. So scientists are very straightforward. They don't, I don't think, take creative writing classes. So as we think about the disease life cycle, the black spot fungus attacks some leaves. Some of the spores get splashed up onto new leaves. The dew's there, it rains 6 hours or more. Okay, the spore germinates, gets in, and then the fungus grows through the leaves. It's called what's referred to as a hemibiotrophic fungus. Hemi means kind of half and half. We're gonna partial. So what happens is it first comes in. It doesn't kill the host cells. It kind of grows between them. It pretends to play nice. And after about day 3, it just goes crazy. It starts killing those cells. Yeah. So then we get this necrosis, the dead cells that turn black, and these lesions that spread. It just goes nuts. And then eventually what happens is that those leaves, sometimes they turn yellow a little bit before they drop. And then they'll fall. And before they fall or after they fall, more rain can splash up spores on additional leaves. and it just keeps cycling all season long. And what we've learned is that the asexual stage of that fungus, it just makes genetic copies of itself without sexual recombination, is what's the typical scenario that we find. So as we grow black spot in the lab on leaves, it's the same genetics that we keep continuing. Nothing gets rearranged or recombined. However, when we're collecting black spot from out in the gardens and around the nation and world, we do find a lot of variation. So somewhere when we're not looking, they're exchanging their genes. And then there's new forms of black spot that have new combinations of genes, some of which might be more aggressive against some of our roses. So we're generally more interested in the resistance of our roses than the fine details of the fungus. At least that's my perception. That's what I love more, the roses more than the fungus, but I appreciate the fungus. So there's two categories of the kinds of resistance that we'll find in our roses, rose black spot, and these same categories also apply to a lot of other plants and disease systems out there, be it our potatoes with late blight and this and that, or whatever it is that you love, your flax and your powdery mildew or whatever. So vertical versus horizontal. So I've been studying with Stan both forms of resistance, but especially the vertical resistance. And a way that really helps at least visualize it, for me at least, is this lock and key analogy. So with vertical resistance, it has an interaction that is like these lock doors and these keys. So a rose might have some resistance genes, and let's say that rose within that circle there on the left has resistance genes A, B, C, and D. Each of those doors represent a locked door. And then the fungus has a set of keys. And that fungus has key A, B, and C, and the doors are like in a hallway. And they can unlock some of those doors, but not all of them. So it can't get in. So our goal is to try to identify as many of those locked resistance gene doors in roses as possible, and cross different roses that have different kinds of locked doors to find their babies that combine as many different doors as possible. So we could really try to lock out that pathogen. The different forms of the fungus that we find out there, we call races of fungi. And what makes a race different from each other is the set of keys. So all the different forms of the fungus that we find with keys A, B, and C, they might have other genes that differ slightly between them, but they all only unlock the same set of doors, and they only infect the same group of roses. So in our work to date, if we found any locked door in roses, it's usually, it only has one locked door. And we've been strategically trying to cross different roses to try to combine different locked doors. So it's at a relatively early stage. And I think part of it is just, it's only the last 30 or so years that people have really seriously tried to breed for resistance in roses. Because they've been spraying their seedling beds looking for the perfect rose, and you can overcome it with fungicides and whatever. So Will Rattler out of Milwaukee, Cordes out of Germany. What they've done over the years, what Will did at his home in Milwaukee, is he'd bring back black spot on all of his trips, and he'd lace his garden. So his goal was to try to unlock all the doors and see what roses ultimately have, what the next category of resistance is, horizontal. So what does horizontal mean? After the rose burglar gets in the house and unlocks all the doors, how much damage can it cause? So some roses, the black spot grows more slowly, so the plant kind of outgrows it a little bit, still looks good for a while. Maybe you're happy when October comes and you're like, okay, you need to rest. Okay, you look pretty beat up, but at least I got most of the summer out of you. So Will would bring home all these different black spotty leaves from his trips. And the goal was to increase black spot and everything basically got a little bit. And then he tried to select those that had less than others. So that's why knockout, when there are keys in the fungus that unlocks it, it doesn't fall flat on its face. It still has good horizontal resistance. So it slows down the progression of that disease. Maybe in your garden, you'll never have a form of the fungus that unlocks any of the doors. Perfect. But if you do, okay, there you go. And that leads to a good message here as well. So sometimes we might, especially years ago, more so than recently, see some advertisements for roses like Baby Love. What's the most disease-resistant thing ever? It hasn't gotten any disease in all of our trials. That's good, that's good. But when it gets to some other part of the country and there is a form of the fungus with the key, after the burglar gets in the house, it might fall flat on its face and look really bad. So really understanding the kind of resistance that we have in our plants. Is it vertical? Is it horizontal? Do we have both? Before the marketers make the big claims, would be really helpful for companies to know and maintain their reputations. All right, so how do we figure out the different races of the fungi? How do we navigate the keys on the keychain, the locked doors? Well, this table here with the little pluses and minuses is how we do it. Along the columns, so those little acronyms, and then there's a number on top of it, a race number associated with it. Those little short abbreviations, like underneath number 1, it's HSN, that's Hansa. The fungus was collected off of the rose Hansa. So we make an isolation of the fungus, okay? We're in the garden in Washington State at some rose garden on this variety of rose. Okay, here's a fungus and we'll grow it. Here's a fungus on this rose here in Farmington. And so that's called an isolate. And then we take these isolates and we challenge a group of roses. And these roses here that make up those rows, those are the names of the roses, are a really good differentiating group. And based on the plus minus patterns in those vertical columns, we designate the races. So if you find a new isolate and it has the exact same plus minus pattern as a previously characterized isolate and race, then you could say, well, that's just another version, just another example of race 5 or race 8. And that's something that I just did recently. There's a postdoc from France working in Dr. Hokinson's lab. And in France, she's been working with an isolate that they collected in France. And we got the permits and such to get it over here. And recently, last 3 weeks, I've been working, inoculating all those roses. And I learned that it's just another isolate of race 8. They were hoping it was a different race, so we could write another paper to get put in libraries where nobody reads it. Yeah. So here's a picture of Will Rattler. There's the original knockout plant, and it was a seedling in 1989, came on the market 2000. Millions and millions have been propagated off that one, and relatives of it with slightly different colors have rounded out the whole series. And there's a new one called Orange Glow coming out next year. All right, and then there's Zach with Will. Zach was one of my first students. And I love Zach. So the first semester I was at school, he was in my woody landscape plants class, and he was a sophomore and was kind of too cool for school and just kind of had an attitude. And by the middle or end of the semester, his heart just melted. He got really excited about plants and I don't know. And then I paid him to help me mulch in my garden the next spring. And then he got excited about roses, and I gave him a whole package of fluorescently colored crossing tags. So we'll know which ones are his, or whatever you want to cross. We'll know which ones are yours. You collect the seeds in the fall. He got all excited. And then he finished his degree. He went to Longwood for a year in their IPM program. And then Will hired him as his rose breeding assistant. And so Zach is the one that made the cross of that Orange Glow knockout that comes out next year. It's kind of fun. All right, so we're taking rose leaves off of rose plants. stripping them off. And those roses are growing in the greenhouse where we don't water the leaves so we don't have black spot on them. So we'll pull the leaves off of the plant in the greenhouse. We'll take it up to the lab. We'll wash them nicely, put them in clear salad containers with moist bounty paper towel, 25 mils of water, just right amount of water. And we'll set the leaves on that. And then we'll put little droplets of water that we steeped in black spot infected leaves. And then we look under the microscope and get the concentration of the spores just right, put the little drops on. And then 2 days later, it only needs 6 hours, but we want to make sure, so we give 48. And then we dry off the little droplets. And then we watch to see, is it a susceptible reaction, like on the left? The black spot grows and little white things pop out to make more spores. Or is it a resistant reaction, like on the right? So this is Dr. Thomas Debner, and he's in Hanover, Germany. And he described the very first 2 locked doors in roses. And we mentioned that scientists are very straightforward in the words that they use. So RDR1 and RDR2, Rosa disease resistance gene 1, Rosa disease resistance gene 2. He described, I love Dr. Debner. Let me tell you a quick story. International Rose meeting in 2013. And he coordinated it, and I was so grateful to go. And we were in this double-decker bus, the first time I've ever been in a double-decker bus. So we went to Sangerhausen in East Germany, this world-renowned rose collection, Rosarium Europa. And so it was really fun. And so our bus driver on the Autobahn, our people drive a little crazy. And so he cut in front of a semi, and he whacked off their mirror and smooshed up their grill, and he kept driving down the road. And then the Polizei came and pulled us all over. And so it was on the highway, and it was out in the country. Okay, so we got off the off ramp. And then, and so it was, sad, and our bus could still drive. But so there's this forest, and us plant lovers started wandering through the forest looking at plants. And Dr. Debner, he's very German, very timely, very orderly, very orderly. And he's like, please stay by the bus. And then maybe he made this up, I don't know. There are ticks out there. So he described the first and the second locked door, but the second locked door is really close to the first one on the same chromosome. And he hasn't talked about it in years and hasn't really done a whole lot with it. So I don't know what's happening with that, but at least Rosa disease resistance gene 1. And this is Vance Whitaker, one of Stan Hokinson's graduate students from about 20 years ago. And that rose in the upper right-hand corner is George Vancouver. And it's an Agriculture Canada rose, part of the Explorer series. And it seems to have resistance in some gardens, but gets black spot in others. So that tells us that there's a locked door that some black spot forms in some gardens can unlock and others not. So Vance started the work to characterize that resistance gene. This is Above and Beyond, and it's nicknamed by some of you in the audience was Zelda when it was in the Umore Park Garden. And I have a bunch of roses to show us here. And I have three little baby plants in there to give away to folks tonight. And that picture on the left was sent to me this morning, and there's Liz. So she's one of my advisees, and she's interning with Will in Milwaukee this summer. And that's his 2-story tall Above and Beyond. So it gets a little bigger in Milwaukee than here. But on the right, is it here? It's here in the Twin Cities area, about 7-8 feet tall. So a good hardy rose. That's pretty healthy. So Vance did his work. He characterized Rosa disease resistance gene 3. We weren't quite sure on what chromosome it was on. But it conferred resistance to a different group of those races or keys than what Debner's RDR1 did. And 15, 20 years ago, there wasn't the genetic resources that there are today. So in about 2014, there was this grant that was funded, this national grant called Rossbreed. And it was meant to fund members of the Rosaceae, mainly fruit crops, like apples and strawberries and things. And the goal was to try to understand the genetics behind fruit quality, flavor. A lot of advancement was made with apples with the University of Minnesota strawberries with people in Florida, California, cherries with Dr. Amy Iazzoni in Michigan, and so on. And they knew about our black spot work and this assay that we have with these little boxes, and it's a smooth, streamlined system to characterize disease resistance in a pretty prescribed way. So we were able to be funded to study disease resistance using some of the newer genetic tools that really leapfrog the opportunity to characterize those resistance genes much more quickly and effectively than just years before that. So what we did was recreate the cross that that Vance did, just because we didn't have those plants anymore after all those years. But George Vancouver was crossed with Morden blush, and Morden blush is susceptible to all the races. It has moderate horizontal resistance, but it gets all the races. And then the goal was to look at the seedlings. So each leaf represents a different, unique individual seedling, and which seedlings were susceptible, which seedlings were resistant to the races that George Vancouver was resistant to. So then ultimately we have all these seedlings, about 100 seedlings. And in this case, about 50% of them had resistance, 50% susceptibility. So then we can dig into the genetics and see what little snippet or region of the genome that the resistant siblings have that the susceptible ones don't. So we could characterize that locked door, come up with a quick genetic marker we could test for with a PCR reaction, and quickly know which seedlings inherit that particular gene. So this cute little thing that you could hold in your hand, it costs about $16,000. and so there's 8 by 12 of those rows and columns there, 96 little blue shiny squares. And every shiny square can test for 68,000 different tiny little regions of the genome for unique sequences. So why is this important? What does this mean? There are things called SNPs, single nucleotide polymorphisms. So DNA is made up of nucleotides, A, T, C's, and G's. And those little letters there in those different rows represents a series of nucleotides. And there's regions of the genome of plants, of animals. We're 99.9% the same, you and I. In terms of DNA sequence, it's that 0.1% that makes us different. So there's a lot of shared DNA. And so we're looking for regions of the genome. See where the letters are red in the middle? It could either be a C or a T. Otherwise, the 35 nucleotides on either side are the same for all the plants. So there are 68,000 different regions peppered throughout the genome where we have a 75 nucleotide sequence where the middle one can vary across individuals. We can think of those like mile markers on the highway, in Highway 94 going from Chicago to Fargo, whatever. And so there's these mile markers. They're spread out quite a bit. And when you break down, tell the tow truck your mile marker, whatever. I mean, there's a lot of space around there, but they could at least zoom in on the region. So that's what this allows us to do. So we'll test every little blue square. We put DNA of an individual seedling and then the parents. And then we compare, we get all this massive amounts of data that takes like big time supercomputers to work with. And then which little mile markers do all the resistant ones share that the susceptible ones don't? And then we can see, well, where does that mile marker land in the genome? There could be hundreds of genes around it. We don't know which gene is the one conferring the resistance, but at least it narrows us in. And if it's a different part of the genome than Dr. Debner's RDR1, we at least know, well, you must be a different gene because you're in a different place. You're a different locked door. So ultimately, we use the system to help us identify where those locked doors are, if they're unique and different, which is really fun. So working with Stan Hokinson, Jim Bradeen, who was the department head in plant pathology, And then Nala Basel and Jason Zern, they're out at Corvallis, Oregon. They did all the heavy duty genetic bioinformatics work for all the different Ross breed projects with all the different fruits and things. And they helped us with the roses. So ultimately, there's just the first page of the paper and a little bit of a table. We were able to identify that RDR3 truly is a unique gene. It's in a different part of the genome. So on the lower left-hand side, RDR1 and 2 are close together on chromosome 1 of roses. Roses have 7 unique chromosomes. And RDR3, okay, it's on chromosome 6. So it takes a lot of effort to find a lot. Patience, a lot of patience. And then Will Rattler, this is his rose Bright Eyes. And it seems really, really resistant to black spot as well. It's only susceptible to race 12. And see that BEP under race 12? The reason why we got that isolated is because we isolated it off of Bright Eyes from a plant growing in Pennsylvania. So a scientist, this is not too creative, BEP. So we thought, well, maybe this has a unique black door too. So we did all the same stuff with those little blue shiny squares. And then we ultimately learned that 68K is a 68,000 SNP array, that axiom array. And so we learned on linkage group 5, which is chromosome 5, RDR4, okay, it's the fourth black door, fourth gene. All right, there we go. And so Bright Eyes only has RDR4, all right. So these roses, George Vancouver only has RDR3. So let's cross George Vancouver and Bright Eyes and find ones that combine both and be able to get greater resistance across more and more of those races. So that's what we continue to work with. So we find these SNPs or these mile markers. Ultimately, though, we want to go a step further. Find, well, what is the gene? Find a little snippet of DNA that we could test for that's within the gene even. So then we can just test for that little snippet with a quick PCR reaction. Does this rose have the resistance or not? Raise a bunch of seedlings from George Vancouver and Bright Eyes. Well, which ones combine both? And we can get rid of the rest. That's the goal. So I made a cross between High Voltage and Lemon Fizz. Lemon Fizz is resistant to all of the races, as we see there in the column there, except race 7 can get it. So that's interesting. So, all right. And so made the cross and we're struggling. We're struggling with the bioinformatics. Lemon Fizz has 3 sets of chromosomes, so it's an odd number. And the genetics is really goofy. I don't know that much about high-powered computer stuff, but the bioinformatics people look at me and grumble, but Lemon Fizz has a great resistance. It's really beautiful. And this rose here is a cross of Bright Eyes that has RDR4 and Lemon Fizz, which we'll call it RDR something eventually when we figure it out. And this rose here is resistant to all 13 races. I tested it. It was bred by my friend Julie. It has a code number. So these grants through the USDA last 4 years. So from 2014 to 2018, and then the money was over. And Stan, there he is on the left, is getting close to retirement. He has some money tucked away. We kept applying for more funding, but for a few years didn't get it. He used money that he had set aside to rent part of the tree nursery greenhouse space, keep the plants going. And then thankfully, this last September, finally another grant was funded that combined studying resistance to rosette disease as well as black spot. And that allowed Deanna, she's second from the left, she's from France, to come as a postdoc. And she's doing some of the bioinformatics work, working with that triploid Lemon Fizz and following up on some of our other work. So the roses are so fragrant, so beautiful, all the hundreds of seedlings over the years as we continue to finalize the final steps and details to come up with a straightforward marker for each of the genes. These are some of the most beautiful Gordon Blush by Bright Eyes babies that have that RDR4 gene. And I hope some come on the market. We'll see. But that one on the left was in bloom today. Ramblin' Red. Ramblin' Red is also another Will Rattler rose out of Milwaukee. And it has a unique resistance pattern. And right before that previous grant that ended in 2018 ended, we were able to afford and get that SNP test done. And then things were just on hold and hold through COVID. Deanna just did the bioinformatics work. And it's on a different chromosome, the resistance gene. It's on chromosome 7. And she just reported it back in January. All right, so we're up to RDR5 now. RDR5. And then this year, out of Baby Love on the left, that's another triploid, 3 sets of chromosomes, awkward. And so I learned my lesson from those bioinformatics people that give me the evil eye. So that orange rose is a baby of Baby Love, and it has a unique resistance. It's resistant to every race except 11 and 13. And then this baby is actually tetraploid. So we used this one instead. Gordon Blush is tetraploid. This is tetraploid. It all worked out. And then Emily, who's a master's student, recognized that it's a unique gene too. It's on the same chromosome as RDR4, but on the other side of the chromosome. So we have these different genes, these different roses that we're finding the genes in. We're accumulating more locked doors. And then ultimately, hopefully have quick tests to help breeders combine them together. So we want to have stuff used. And so that's a challenge. So as we come up with these markers, working with diagnostic labs that would do the testing for a service, because a lot of breeders, nurseries don't have a PCR lab in their bedroom or whatever, side office. So but we want to make this knowledge accessible. So finding as many locked doors as possible. Julie's Beautiful Seedling is a great example, combining RDR4 and whatever is in lemon fizz that eventually will number. There's Deanna, and there's the name of that new grant that's funding Deanna's postdoc. So she loves spending hours at the computer. She knows how to program computers with that code stuff, not that point and click. So I'm really grateful. So here's an example of oodles of boxes that we're gonna put rose leaves in. Just those little salad boxes. We actually get them from restaurant supply companies. All right, so the same process with any kind of plant disease. Find segregation of resistant individuals, susceptible individuals. It doesn't have to always be disease. It could be double flowered versus single flowers, whatever trait that you want to follow. And then use something like that SNP array, those mile markers, zoom into that region, see what genes are in that region, narrow down your search, find what you're looking for, leverage it to more strategically do selection for the apple breeding program, for scab resistance, for the fruit qualities. It takes an apple from seed. If you don't graft it on a precocious rootstock, it could take a decade or more to flower as a longevity period. So these DNA tests at least help us whittle down the population to hopefully at least pre-select those that we know will be crispy or have whatever it is that we want. And then out of whatever we save and raise to maturity, then we could look for those additional traits of value as well. Thank you.
CRISPR technology to pull out the pieces you want and stick them in the plant.
Good question. So the question is, can we use CRISPR technology to be able to come up with targeted mutations? So it's possible. Alan Smith at the University of Minnesota is using CRISPR to try to help fight invasive plants like tansy right now. So right now, it sounds like that there aren't very many regulations, at least in the United States. In Europe, they think of this targeted mutation as a GMO, and it falls under all the regulations. So we'll see if things change in the US. So supposedly right now you're not taking foreign DNA and stuffing it in. You're just tweaking the sequence, moving it, tweaking, adding a little bit, deleting a little bit. Yeah, so it's really challenging. There's a company called Calyx, and they're in Roseville, and Dr. Voitas, he's part of a group that owns it. And so he's trying to tweak alfalfa to make it more digestible, as some patents, and tweak this. And I don't know, it's not going as smooth as he's hoping to, or at least business wise either. And so we'll see. We'll see how our people take it. Maybe we could just tweak roses and make the locked doors take a sequence.
I was going to say, if you're going to take plants and breed them to get what you want, it's not that much different than if you're taking this piece and putting it in that plant. In the end result, the same.
Yeah. So yeah. So some people, I think to a large extent, I agree. Yeah. So Dr. Bradeen, he worked with potatoes, and there's a region in Solanum bulbocastanum that has great late blight resistance. And it's a real difficult species to cross with modern potatoes, and that really goofs up the tuber qualities. So his goal was to try to uniquely cut out that region without adding the antibiotic resistance, selectable markers, and that other tools that make it easier to find what you're looking for. And then just say, well, it's potato to potato, and maybe it would have greater acceptance. Yeah.
Hi, I'm over here.
Hi.
Oh, you didn't know where I was. I don't know about the rest of the people in the room here, but so much of this was over my head. It's like I was just swimming with information. It's fantastic, but I just wanted to say thank you, because when we go to the store and we look at the rose bushes and we go, why does that cost so much? This is why. There is so much research that goes into this to give us, what we perceive as the perfect rose, the perfect bush, the perfect smell, perfect color, whatever. So all that you're doing is what goes into that cost. And I just wanted to say thanks. And you present it really well. It makes it fun. Thank you.
Thank you. Thank you. There's a lot of plants that people go through that don't look that great, that take a lot of care, a lot of resources to fertilize, irrigate, labor, and to get to those that that push the knowledge forward.
I was really interested in how you described your genetic research and all the work that you're doing to get the disease resistance and stuff. But a lot of people are kind of going back to the heirloom plants.
Yeah.
And, like those were because a lot of roses were bred for smell, or they're bred for color or uniqueness or whatever. But there's all this talk about, heirloom plants and stuff. Can you give us a perspective of heirloom versus all the work that you're doing, and how that, and what your thoughts are on that?
That's a great question and perspective. So with that RDR3 out of George Vancouver, I looked at George Vancouver's relatives, predecessors, and what I've been doing the last few years is gathering a lot of ancestors of George Vancouver and relatives and older roses to try to learn, where did that gene come from? How did it go through the generations? What's its story? And so far, I was able to trace back the two oldest roses, Goldilocks from 1945, and then Pink Pet from 1928, have the same resistance pattern, and I think the gene. So as we think about heirloom varieties, our more recent varieties trace back to what we had before. And so a lot of these genes, they're not really new. They've been somewhere before. And so with germplasm repositories and saving older varieties, making them available, those are great tools for breeders. Like for instance, for my master's, I studied late blight resistance in potatoes. And I screened these wild potato species native to Mexico. There's a new form, a race of late blight that came out in the 1980s out of Mexico across our region, parts of the world. And so because people saved those wild species as well as some of the heirloom varieties. I was able to screen them and see who had resistance so we could go back to go forward. So I value heirloom roses, heirloom of whatever. And they're beautiful, they're unique. Sometimes at a visit before here tonight that some of the heirloom varieties might not have resistance to the current diseases we face. Maybe the diseases at that time, they were great, but just because diseases are dynamic, you know, they might struggle today. So, oh, you're welcome. That's a great perspective to think about. I love that. Yeah, yeah.
Yes, I do.
Great presentation. I really appreciate the information. As you were talking about other species or going up the family, genus, species, all of that. You think about like crab apples and other things. Is black spot only roses as we think of versions of roses, or does it go further in the Rosaceae family?
That's a great question. So as far as we know, diplocarpon rosea is obligate. That means it only affects roses. There's other diplocarpon species that affect you know, other types of rosaceous crops, but that specific species only infects roses. Yeah. So, but some pathogens have a wide host range and some are very narrow. Yeah. So that's interesting. So one of the challenges, here's a perspective for disease that at least I find really interesting and helps guide some of our recommendations for diversity spreading risks, especially when we think of the urban forest. It's those species that are within the same genus, like for instance, elms in the genus Ulmus. There's elms native to North America, to Europe, to Asia. When we find a group of plants within the same genus with a wide geographic range like that, we're more vulnerable. So when there's a fungus that affects, for instance, the elms of Asia, and then they've developed at least enough tolerance or resistance because they didn't go extinct, and then people move them to Europe, and then their European elms were going down hard, and Dutch elm disease. And then the fungus came over here, that our American elm. And same with chestnut blight. So these big pandemics, emerald ash borer, like in Korea, the little insect, it didn't do much damage. Those ash are pretty good. They didn't, you know, die left and right. They otherwise would have been extinct. And then that little insect comes over here, and here's green ash and white ash and a smorgasbord. So that's a good perspective. So if there's a lot of close relatives around the world, we should look even more closely in terms of what diseases are out there and how they're spreading to help reduce risk. So for the urban forest, the idea is the 30-20-10 rule. You don't want to have more than 30% of your trees within the same family. You don't want to have more than 20% in the same genus. You don't want to have more than 10% of the same species. So this rose here is a hybrid of Rosa arkansana. So when I did my master's with potatoes in the late 90s in Morris, Minnesota, we had a research station there. And the people with me in the van were very accommodating. I said, oh, stop the van. And so I dug up a really beautiful specimen of Rosa arkansana. And then this one is a hybrid. And so this one's really pretty and was in bloom today. And then talking about heirloom roses, this is Thérèse Brunet from the 1950s. And so it's really, really hardy, real fragrant. It has some of our native Rosa blanda in it. So an heirloom is anything that's over 50 years old. And you mean, yes, yeah, me too. And that we're not a hybrid or this or that. So if you grow a bunch of it and just save seed amongst itself, it comes relatively true. So beautiful, beautiful hardy rose, Thérèse. This rose here is thornless. It's Pax Apollo, and it was bred by Niels Hansen. And Niels Hansen was at South Dakota State University. in the late 1800s, early 1900s. And I'll share this quick story just because it inspires me so much. So Mendel did his work with sweet peas in the 1860s or so, as papers came out, mid 1860s. And they were just in a dusty old library. People didn't read them. And then about 1900, different research groups around the world were stumbling on this idea of discrete units of inheritance and, genetic transferability across generations. Before that, people thought of genetics like mixing paint. You got a glug of mom, a glug of dad. You got more mom, you got more dad. Look at you. And so just kind of mix it together. And so these discrete units of inheritance and categories was unique. So not everybody believed in genetics at first. There was Zlesak and all these stories and people starving in Russia. And so Niels Hansen made the huge trip to the very first genetics conference in London of 1905. And he brought back in the late 1800s, 1897 or so, hardy versions of alfalfa from Russia, hardy fruit trees, pears. He brought back Rosa laxa, which is the very grandparent of my above and beyond that's so hardy from Siberia. And so he has a huge fingerprint on genetics and of our hardy plants, not only of agronomic crops like alfalfa, ornamental crops like roses, fruits too, because his goal was to try to enrich the agriculture of the upper Midwest. And so it's been really fun to get to know his great-granddaughter, Jean Peterson. She did her master's looking at the economic return on investment in supporting publicly funded plant breeding programs and using your great-grandfather as an example. So years ago, I got Pax Apollo from a lady named Wendy in South Dakota, and I kept it going. And then I shared cuttings with Jean, and then Jean got some of those plants at McCrory Gardens there at South Dakota State University in this wonderful garden to honor Niels Hansen. So there's heirloom roses here, there's modern roses. So what I have in that bucket are 3 young little plants of above and beyond, and then 3 bigger plants of these polyantha roses. Polyantha roses, poly means many. So there's many little flowers on these roundy moundy bushes. Here's one that I like that's kind of purple with a white base of the petals. So there's 3 of those in those bigger pots. And then above and beyond, even though they're real little, how big they get. So I'll tell you a really quick story. So we'll give those out to people here. We'll find a way to get them to you. But years ago, there was a wonderful master gardener. I hope she's still doing okay. I haven't visited with her in years. Naoko. She lives in Washington County. And she used to go to Linder's and just buy oodles of plants. So many plants. She just loved plant oodles of things. And she came over to visit me in St. Paul. And when I lived there, and I had a wonderful landlady who let me garden. And Naoko, she's Japanese. She came over and she told my landlady, Marianne, even gives me these little roses. They're so little, but then they get so big. They did. Above and beyond and its relatives, it's true. So they start off so little, but then they'll get so big. It's a very insightful question. So the question was, does the amount of repeat bloom affect health and disease resistance and durability? So as the plant's putting energy into these flowers that are kind of a dead end, right? There's no chlorophyll in those petals, typically. So what we found in segregating populations where repeat bloom is typically recessive, and you have these siblings, some of which repeat bloom strongly and some that don't. The ones that do repeat bloom strongly do tend to get more black spot. And then they also tend to be a little less winter hardy because they're continuing to bloom versus hardening off and slowing their growth down to get ready for winter. Yeah, so resource allocation is really important.
Thank you.
You're welcome. So above and beyond, it blooms really great in the spring before the Japanese beetles come. And then what happens is that it'll repeat bloom a little bit through the summer. And then what happens is that it knows when winter comes and it shuts down. So that's why it's so hardy and can stay so big.